Okay, this is then the Inheritance and Selection knowledge test. Okay, there's a fair few words in it, keywords, decent bits of information. Right, so we need to absolutely focus on getting all the words correct, because obviously I've said before in a biology exam, right, in particular, you must use the correct terminology to get those all important marks. What shape is DNA? DNA is a double helix. Wow, I'm writing big there. Okay, so it's a bit like a spring, right? And then it's kind of, I'm terrible at drawing this, it's kind of then got another spring that runs along with it. Right, so it's like a double helix. What is the genome, and what are the three main medical reasons why we study it? Right, so what the genome is, is a complete set of DNA. Right, and that includes the genes. Right, so it's every single kind of human strand of DNA and genes, it's all kind of put together, right, into the genome of humans. The main reasons why we may well study it are, what we can do is we can look for genes linked to, how many E's has linked got? Linked to disease. So what we can do is we can find out that cystic fibrosis is on chromosome number 7, and it's having two faulty genes, or two faulty chromosome number 7s that then give you cystic fibrosis. Another one, it could be understand and treat inherited diseases like cystic fibrosis, like Down syndrome, right? It gives us more of an idea about what's actually going on. And the third one is trace migration. So what we can do is we can see where the genes or the genome within your own population have originally come from. They could come from Asia or India or Sweden or wherever it may well be. Okay, question number 3, comparing asexual and sexual reproduction. So sexual reproduction has two parents, right, mummy and daddy. Asexual reproduction only has one parent. Sexual reproduction is the offspring are non-identical. Asexual, they are identical. All right. Now, what you can actually see is, you can see here what I'm doing is I'm doing fact by fact. I'm doing kind of like a comparison between the two. Sexual uses meiosis, and asexual only uses mitosis to produce the offspring. Okay, so meiosis is then that process where you're producing your gametes, your sex cells, where you start with 1 and you end up with 4 with half the number of chromosomes. Based on that, mitosis produces gametes, which is your sex cells, which then leads to the fertilization process. In asexual reproduction, there are no gametes. Okay, so basically what they're doing is they've just got 1 and it kind of divides into 2 and it keeps on reproducing like that. There are also 2 that then go in the middle, right? You will make a new individual, and the fact that in both genes are passed on. Okay, but it's a good comparison question. And what you do is you just need to think about number of parents, identical, not identical, how does the actual process actually happen, meiosis, mitosis, whether it's got gametes or whether it's not got gametes. Okay, and it's good like a comparison, and just do one and then the next, one and then the next, rather than doing as a list. Question number 4, right, this is then mitosis and meiosis. Right, these are then 2 words that you must, if you get them in an exam, you must spell them correctly. Okay, so mitosis produces 2 cells. Meiosis produces 4 cells at the end of the actual process. The number of chromosomes you get in humans, you'll get a full 46, and in meiosis, you'll get 23, which is then the sex cells, okay, sperm and your egg. The genetic comparison is all of the things, right, are identical. In this one, there is variation, which means that there is differences between them. Question number 5, if B is dominant and big B is dominant and little b is recessive, what are the possible combinations and what are they called? Right, so I'll start off with big B, big B. All right, now because the two are the same, that is homozygous, and then because there's two big ones, dominant. Another combination, I'm just going to do little b, little b. Because they're both the same, homozygous, and because they're little, recessive. Okay, kind of means they're not as strong. And the final one is big B, little b, which is, heterozygous. All right, heterozygous means you've got one of each. Question number 6, complete the Punnett square and what are the percentages of each? Right, so if I got big B, I'm just going to put big B, because I've got that one and I've got that one. Then I've got big B there and I've got big B there, so I put big B, big B. I've got big B, little b. Oh, should I change my colour? That's exciting. Big B, little b. And then I've got green, I've got big B, little b. Alright, what you can do is you can work out that there's 25% of that, 25% of that, 25% of that, and 25% of that. But how much big B is there? Big B and big B, 25 add 25 is 50%. If you've then got big B, little b, the percentage of that is 25 and 25, which is also 50%. What call is polydactyl and cystic fibrosis? Polydactyl is great, right? Absolutely love it, right? So what it is, it's caused by, right? It's caused by a dominant allele. Dominant allele. All right. So what that really kind of means is if you've got a chromosome from mum and a chromosome from dad, and the polydactyl is that one there, right? And then dad doesn't have the polydactyl chromosome, because the yellow one there is dominant, that one will then be from mum, and mum will then allow or give the person polydactyl. And what they have is they have extra finger or toe, okay, or both, or even more, right? They could have 7 fingers or 7 toes. Cystic fibrosis, right, is caused by a recessive gene on chromosome number 7. Right, so what we've got here, and I'll just draw my random picture again, right? I've got chromosome from mum, chromosome from dad. The red is the cystic fibrosis. Okay, and then I'll just do a different colour, or lots of different colours. The yellow is not cystic fibrosis. So if you've got a chromosome from mum and a chromosome from dad, right, and dad has given you the non-cystic fibrosis, and mum has given you the cystic fibrosis, which is the red one, you don't have it because the red cystic fibrosis chromosome is recessive. The way you do get it is if you've got chromosome from mum, chromosome from dad, but mum gives you the recessive and dad gives you the recessive, then you do have it. Okay, and these two different versions of the same chromosome are called alleles. Symptoms, right? I mean, there's quite a number of symptoms, right? But what I'm just going to do here is I'm just going to write down excess mucus, right? So the mucus can be in the breathing system, respiratory system, or the digestive system. Question number 8. Okay, What is gene therapy and what are the three main issues? Gene therapy is replacing a faulty gene. Okay, now if you imagine, right, if you've got trillions and trillions of cells in your body, to actually replace a gene within is actually incredibly difficult. All right, and there is three main issues. I'm just going to write down here, economic. Okay, so what it means is it's a very costly process and it is not 100% effective. Number two, social. It is not available to all. Okay, if you think it's very, very expensive. And number three is ethical. Okay, so ethical, right, should only healthy embryos be implanted. Okay, so what we're talking about here is you've gone to gene therapy, you found out that your embryo has got faulty genes in it, and so what you do is then you can then replace those faulty genes in the embryo. All right, is that the right thing to do? Is that fair that what you've done is you've gone in and changed the genetic makeup of that embryo? All right, and ethically, is that the right thing to do? Question number 9. What are the two main causes of variation? We've got genetic, and then we've got environmental. Okay, so genetic means, right, that obviously if you've got a brother and sister that come out at different times, they will be genetically different, even though they've got the same parents. Environment, right, is where you might have two identical twins where their genes are exactly the same, but one gets taken to South America, one gets taken to the Arctic, right? How are they going to be different due to their environmental situation? Question number 10, speciation. This is then where a population can be split into different species, right? So, I mean, in a very sort of basic kind of way, a species is an animal that can reproduce and produce fertile offspring. If the population gets a little bit too diverse, that then the animals within it, or two animals that are then genetically even further away from each other, cannot produce fertile offspring, speciation has happened where you've then created another species. Best example of this ever is the Galapagos Islands, right, where what you've got is you've got Galapagos finches, where they've been apart for such a long period of time that then they have done speciation, which means that they then cannot reproduce between each other. Question number 10, or 11 rather. Right, question number 11 is list 5 reasons for a species to become extinct. Now there is lots and lots and lots, right? You could have better adapted competition, right? And the best example that comes up a lot of these times is the grey squirrel and the red squirrel. Right, so even though the red squirrel isn't extinct, the grey squirrel is better adapted, it's bigger, it's stronger, it's more aggressive. So what that can do is the competition is then winning over the red squirrel, over the food sources. Species to become extinct, disease. All right, there was a disease quite a large number of years ago called myxomatosis. And what myxomatosis did, it was a new disease and it wiped out rabbits. And it killed something like 99% of the rabbits in Australia. Right, and what that then did is it was used by us to actually try and get rid of the rabbits. right? But a disease in itself could appear that then makes animals extinct. Human predation. Okay, good example of that could be the dodos, right? Dodo, big bird, right? A lot of it will have then been due to dogs, rats, cats eating eggs, killing the birds themselves, but it's also the predation of humans. And what humans will do is they'll go out there and they'll shoot and eat, right? Or even just go and shoot for fun. Fourth one rather, is lack of food. If there is not the right type of food in a certain area, a species could become extinct. We've also got invasive species. That is then where a species that isn't naturally in somewhere that then manages to make its way in. So again, like rats in Australia, it's an invasive species, and what it does, it eats lots and lots of the food that the native animals then would want to be eating. There are others as well, right? You could also have climate change. You could also have loss of habitat. Okay, so because humans are so clever, chopping down things like the rainforests, right? What we're doing is we're destroying species, right? Potentially making them extinct by taking and chopping down their habitats that they live in. Question number 12. What is selective breeding? It's choosing parents with desired characteristics. Parents with desired characteristics. Right? So it could be the way an animal looks. Right? If you think about pugs. Pugs 200 years ago didn't look like pugs they do now, but what they've done is they've been selectively bred to make them look like they do. And exactly the same thing with animals, plants rather. What you can do is you can select a plant that produces more potatoes and selectively breed it to bring on the best and most desired characteristics. Process of genetic engineering. Right, so the first stage is enzymes are used to isolate a gene. Can't fit it in. Then what happens is the gene is inserted into a vector, right? And a vector could be a plasmid, right, or it could be a virus. Then the vector delivers the gene. And the final stage of it is new characteristics develop. Okay, so now the actual genetic engineering process is quite complicated, right? So it'd be worth just kind of looking up that into a little bit more detail than what I've then just done. But those 4 stages are the main kind of principles. Question 14, what is genetically modified crop and what are the 2 main advantages? Right, a genetically modified crop is where you've got genes from other organisms. Okay, 2 big advantages are those new plants might be resistant to insects and herbicides. And the second one could be you might increase crop yield. So what people are doing in their genetically modified crop is where you're going to get genes from another plant, right? And what you've got is you've got plants out there that are resistant to insects. And if you find the gene that is where it makes them resistant to insects and you put it into your potato crop, then they will become resistant to insects as well. And you can also find genes that will increase the actual yield of the crop. Question number 15. What is an antibiotic resistant bacterium? It is basically a bacteria that is resistant to all antibiotics. Best example of that is MRSA, which is methicillin resistant Staphylococcus. Two there. All right, and what that is, so that's a bacteria that is resistant to antibiotics, right, and it is horrendous if it actually gets into a hospital in any form of way at all. MRSA. 16. What is the Carl Linnaeus classification system? Right, now, a mnemonic. Now, this has been on exams possibly a couple of times in the last 3 years. Right, so the mnemonic is King Philip came over for green soup. Okay, and why not? Why wouldn't King Philip come over some green food? One I used to do when I was younger was King Philip came over for 5 giant sausages. Right, which is similar, but this is quite a good one. You kind of remember it. So King Philip came over for green soup. Now, what that actually means is the classification, that is the kingdom. Okay, that's the kind of the big group. Then what it does, it kind of breaks down a little bit. It goes to phylum, then it goes to class, then it goes to order, then it goes to family, then it goes to genus, and then it goes to species. Okay, and every single living thing, right, on the whole of planet Earth fits into this kind of process, right? So humans, we are, Animalia. Okay, so we are Animalia, that's an I there rather than an A, right? Animalia, right, so we're animals. We are then in the Chordata phylum, which means we've got a backbone. We are then Mammalia, which means we're mammals. We are then primates. We are then Hominidae, which is the kind of the next name, it's like the family that we're in. We are then Homo sapiens. Okay, so that is our full breakdown based on the Carl Linnaeus classification system. Right, but what you need to do is you need to remember this mnemonic here, right, so that then what you can do is you can remember this. They could choose any example of any animal ever, right, but what you've got to do is you've got to be able to pick out what they're missing, because they'll usually be something like a gap fill. Question number 17. What 3 is a fossil? Well, let's get rid of that 3. What is a fossil and what can fossils consist of? So fossil is the remains of an ancient organism found in rocks. Okay, so they can be parts not decayed, they could be parts replaced by minerals, and they could be preserved traces. Okay, so examples that we could maybe have. Parts not decayed, right? In Russia, they found a woolly mammoth, right? And it was into the permafrost and it had been frozen very, very deeply for a long period of time. And they found almost like real hair. Parts replaced by minerals, right? That is then your typical fossil that you may well think of, where you see the pattern of it in a rock, where the actual structure of it has then become crystallized. And preserved traces could be footprints, right? Where there are certain places in the world where there's like dinosaur footprints. Question number 18. What are the three main parts of the domain system, right? So what we've got is, we've got archaea, we've got bacteria, and we've got eukaryota.